So next we'll look at the ER. Again, let me clean this up and redo it. Okay. Cell, nuclear envelope, extension of the nuclear envelope with all these channels. This is our nucleus here. So this extension of the nuclear envelope with all these channels, this is called the endoplasmic reticulum. So the endoplasmic reticulum is it's a series of channels. Again, it's a layered membrane that's continuous with the nuclear envelope. So it's involved, well, it has two parts. So whether you're looking at it, when you look at it, if you see all these dots on it, those are ribosomes. So this is going to be the rough ER. It's rough because it has all these little balls in it, these ribosomes. And then you have smooth ER. Let's talk about the difference of what is being made in each. Okay. So smooth. The first thing, easiest one to remember, S. So let's put another S. S. This is going to be synthesis of another S word, steroids. So if you see the word steroid hormone, testosterone, anything like that, that is going to be, or he could say something like fat-soluble hormone, because remember, steroids are a lipid, lipids are fat-soluble, so just trying to show you all the ways that he could ask this question. But if it's a synthesis of a steroid and he wants to know what organelle is responsible for that, that is the smooth ER. All right. The other things that it's involved in is the metabolism of lipids and the metabolism of glycogen. I don't have great ways to remember those. You kind of have to do these through repetition, but I do have pretty good ways to remember this rough ER stuff. So the rough ER, remember it has those ribosomes on it. Ribosomes are the site of protein production. So what kind of proteins are made here? Export proteins. Remember we said free ribosomes make things that are going to make proteins that are floating in cytoplasm. Attached ribosomes are making export proteins. So export proteins. It's also doing phospholipids, creating phospholipids, and integral proteins. Let me show you a picture that I think really helps nail this on the coffin. So nucleus, extension, blah, blah, blah. This is my ER. These are all my ribosomes. Okay, so what's leaving here? We already said export protein, so I'm just going to put export protein. I'm going to draw an arrow that shows leaving. We want to produce other things that are going to go in this general direction. The difference is that they're not going to cross that plasma membrane. So we need things that are approaching the plasma membrane and being integrated in with it. What's the most densely populated component of this plasma membrane? Phospholipids. So here we see that the rough ER is making phospholipids, and that is going towards the plasma membrane. What are the proteins found within that plasma membrane? Those are integral proteins. So once again, we have something heading towards looking like it's leaving, but this time it doesn't. It just gets integrated with the plasma membrane. So we have export proteins, we've got phospholipids, and we have integral proteins. Those are the three things the rough ER does. All right, this one's pretty easy. The Golgi apparatus, he calls it the UPS store because it does a few different things. So it's going to modify whatever that came out of the ER. So it's going to modify proteins, but it's also going to concentrate them, which are these brackets, and then package them to leave for export. So it takes these things from the ER and puts it into a vesicle. We talked about a vesicle being this ball of phospholipids if you've been staying up with these videos. And then finally, lysosomes.